Classification of enzyme. Enzymes are biologically active proteins that catalyze chemical reactions in living organisms. They act as catalysts by lowering the activation energy required for a reaction to take place, thereby increasing the rate at which the reaction occurs. Enzymes are highly specific, meaning that they typically catalyze only one particular reaction or a small group of related reactions. Enzymes are typically classified into 6 main categories, based on the type of reaction they catalyze. Oxidoreductases. These enzymes catalyze oxidation-reduction reactions, such as the transfer of electrons from one molecule to another. An example of an oxidoreductase is the enzyme dehydrogenase, which catalyzes the removal of hydrogen atoms from a substrate. Alcohol dehydrogenase is an example that converts ethanol to acetaldehyde. Transferases. These enzymes catalyze the transfer of a functional group from one molecule to another. An example of a transferase is the enzyme kinase, which catalyzes the transfer of a phosphate group from ATP to a substrate. Glucokinase, an enzyme present in the liver and pancreas, transfers a phosphate group from ATP to glucose and helps in the metabolism of glucose. Hydrolases. These enzymes catalyze the hydrolysis of a substrate, which is the process of breaking a bond by adding a water molecule. Examples of hydrolases include the enzymes that break down sugars and the enzymes that break down fats. Lipase, an enzyme found in the pancreas, helps to break down fats by hydrolysis. Lyases. These enzymes catalyze the cleavage of a substrate by means other than hydrolysis or oxidation. An example of a lyase is the enzyme that catalyzes the conversion of fructose 6-phosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Carboxylase is an example of a lyase enzyme. It catalyzes the conversion of pyruvate to oxaloacetate. Isomerases. These enzymes catalyze isomerization reactions, which involve the conversion of a molecule from one isomer to another. An example of an isomerase is the enzyme that catalyzes the conversion of glucose 6-phosphate to fructose 6-phosphate. Phosphohexose isomerase is an enzyme involved in the isomerization of glucose 6-phosphate to fructose 6-phosphate. Ligases. These enzymes catalyze the formation of a bond between two molecules, usually by the hydrolysis of ATP or a similar high-energy molecule. An example of a ligase is the enzyme ligase, which joins two DNA strands together. In summary, enzymes play a vital role in many biological processes. They help in the metabolism of nutrients, detoxification of harmful substances, and DNA replication. A dysfunction or deficiency of specific enzymes can lead to a variety of diseases. Understanding the specific function and mechanism of enzymes can help in the development of new drugs and treatments for these diseases.